Well, dear students, we will continue our course of molecular biology, and today we will be tell about transcription and translation of genetic information. Now again, all described by central dogma was proposed by Francis Crick. DNA is storage information about proteins. RNAs, it or carrier of information, or can be end product, for example, ribosomal and transfer RNAs, and the proteins as a result of realization of its information. And all information described on pieces which call genes. No, it typical demonstration, a basic unit of heredity, but really it's segment of DNA or RNA or RNA viruses, which consist information about synthesis of biological active products, usually proteins or RNAs. And distribution of genes, we talked about it on previous lecture, distributed uneven, not randomly, but it reflects evolution of genome and expression of genes. You can see on this plot more or less tightly and more or less intensively. And the number of genes, you can see a number of genes in different chromosomes, really 22 pairs of autosomes and 2 sex chromosomes. And now you can see number of genes and length of chromosomes. Chromosomes consist from any tens to any hundreds base pairs nucleotides, and you can see very long first 2 chromosomes and X chromosome. It more than 150 millions nucleotides, and the number of genes, for example, first chromosome, first, second, 11th, 12th, and X chromosome, density of genes relatively high, up to more than 4,000 genes in first chromosome. But usual average density of genes, you can see 12 genes on 1 million base pairs. But of course, it's very uneven. And now you can see distribution of genes and sometimes the candidate genes, sometimes maybe pseudogenes. Distribution in sex chromosomes. The chromosome, for example, X chromosome, consists a lot of genes which will be distributed relatively tightly, and Y chromosome consists individual sex-dependent, male-dependent genes associated with one part of chromosome. Big part of chromosome gradually decrease, by the way, because absent of recombination. Recombination between X and Y chromosomes is absent. And an organization of genes, you can see not sample, but example of distribution. In any place of chromosome present a lot of parts which can be described, stained by special staining, again by different places, by different colors, more or less intensive content of purine and pyrimidine bases. And now you can see distribution of genes. Sometimes it can be separated one to other, but sometimes maybe overload one to other. And you see usual gene consists near 30,000 nucleotide parts, but proteins usually consist some hundreds amino acids, and in folded state it will lead to functional active structure. Now with real example of gene, now with, for example, nitric oxide synthase 3, it enzyme which will be regulate intensity tension of wall of blood vessels, and you can see consist, if you will calculate, introns dark greens, but which will be exons participate in protein structure, but a lot of light green exons with absence of not participate in protein structure, and will be excised after transcription. And you can see overload of different genes one to other, but it nonsense parts of genes, again exons without translation, transcripted but not translated. And now it is spacer between nearest potassium channel and any other gene, but it chromosome 7. You can see end of it chromosome and indication localization 7q36.1. What is it? It indicates about all chromosome divided on two parts, separated by centromeres. Short arm called P, letter P, a long arm called letter Q, and different distance from centromere have different number, different digits, and 7q36.1 is 7th chromosome, Q a long arm, and 36 you can see 6.1. It localization of it marker of it gene in this part. Now you can see this orientation of genes, orientation of coding part can be coding and template part can be oriented in different parts of DNA, because DNA is double strand, and one strand called template, which is used for synthesis of messenger RNA, and second strand called coding. It consists sequence of nucleotides, correspond sequence of nucleotides in messenger RNA, but instead of thymine will be uracil. And now you can see in viruses overlapping genes, but in human genome you can see also overlapping take place also, but it not frequently and not functionally active. But sometimes maybe very interesting variants, but about it we will tell some later. Now it length of proteins, number of amino acids, distribution of proteins, length of proteins, beginning from some tens amino acids, maximal number at from 200s to 500s main peak, and gradually decrease, but any proteins in our cells consist number of amino acids up to 2000. Usually it membrane channels. But no, any variants also take place, maybe. Now we will tell about it more detailedly. Now you can to see length of human genes. If length of amino acids some hundreds, length of genes much more. But sometimes any genes correspond length of any genes correspond length of amino acids multiplicate on 3. For example, tRNAs without inner space, without inner introns, and number of nucleotides correspond length of amino acids. It intron-free genes, tRNAs, histones, alpha interferons. But now it percent of sequence of gene sequence in introns. And you can to see a period introns and length much more increase and increase. It kilobases length of genes, and it achieves some hundreds, sometimes some thousands, and the maximal length of genes you can to see there. It more than 1 million nucleotides, and most elongated gene is dystrophin, which consists up to more than 2 millions, 2.5 millions nucleotides. It's very big part of correspondent chromosome, by the way. And looking please genes which coded structure of immunoglobulins is very long. Second and third place in this comparison, it light chain of immunoglobulins and heavy chain of immunoglobulins. It's important for maturation synthesis of immunoglobulins with multiplicity of active sites for recognizing of antigens. We will be tell about it again some later. But dystrophin, it muscle fibers, component of cytoskeleton, very interesting and important protein, and mutation in this gene lead to myodystrophies with serious consequences, serious pathology. Also, cystic fibrosis transmembrane conductance regulator, also very big, some thousands, 100,000 of lengths, and mutations in these vectors can be relatively frequent. Now it summary of it comparison between length of genes and it products. Now it's 24,000, really now 22,000 genes, genome. Average size of gene, it's 27,000. Maximal length, largest gene, it 2.4 million nucleotides. Shortest, you see transfer RNAs. Largest exon, 7,500 nucleotides. Average exon number 9. Largest number of exon in titin. Titin is spring protein in muscles. 363 exons. Frequent can be mutations also. Largest introns up to 1 million. And largest polypeptide, titin. And genes without introns, it mitochondrial genes, RNA genes, and interferons. Again, picture of different groups of genes which coded different groups of proteins. We talked about it on previous lecture. You can to see a lot of genes which coded take coding of different groups of proteins. Any proteins must be synthesized every time. Genes for it, proteins called housekeeping genes. Any genes will be activate expression in any special conditions. Genes will be activate depend on situation, and a very big part of it genes, some less than half, it genes with unknown function of its products. Again, expression of any genes, synthesis of messenger RNA, take place with different intensity, and products of it transfer RNAs can be different concentration of transfer RNAs, and again result of translation can be different. Why it depends? It depends on the intensity of transcription, and it depends on frequency of possibility of synthesis of each gene by correspondent enzyme system. No, by the way, convenient case for repeating of different types of RNAs. It messenger RNAs, individual for each protein. It tRNAs, different hundreds variants take place, but all grouped in correspond 20 amino acids, trans-RNAs. Ribosomal RNAs. Ribosomal RNAs structure and provide structural and functional properties of ribosomes. Also, small nuclear RNAs. It important for excision of introns, splicing, and microRNAs. RNAs for regulation lifetime of messenger RNAs. And translation into proteins, only messenger RNAs, and it depends on its quantity, depend on activity of correspondent genes. About it some later. Now what we can to see when we will be see on template one of double strands, template strand of DNA. It depends on its sequence. Really, if you will be see on its sequence, we cannot to see anywhere. But gene will be present there, and we can to recognize it if we will be compared and check it sequence of nucleotides. And what parts of genes can be present. It of course must be coding sequence which will be important for its sequence realized to amino acid sequence in proteins. But it will be beginning, by the way, by start codon and finished by so-called stop codons, correspondent sequence of nucleotides. But it must be upstream of it, start of synthesis, transcriptional start, without it will be upstream before start codons, and place for binding of enzyme which will be synthesized messenger RNA. It's enzyme called RNA polymerase, and its place of its recognizing must be localized upstream of transcription start site. Promoter with corresponding specific sequence of nucleotides. For example, in prokaryotic promoters must to have special sequence of nucleotides, will be high rich thymine and adenine, TA box, so-called, no, especially it's important for eukaryotic, it called TA box. For prokaryotic, it specific name, it Pridnov box, so-called. It -10 upstream of TC transcription start site and -35 nucleotides. Sequence of it important for recognizing of RNA polymerase. And also after finishing, after stop codon, must be specific sequence which will be indicate about finishing of its gene. Sometimes it may be a specific sequence which can create stem and loop, so-called terminal loop. Well, again, it repeating one strand template strand orientate from 3 to 5 prime end. You see, from 5 to 3 prime end, complementary antiparallel strand call coding with sequence which correspond messenger RNA. Well, next important differences between pro and eukaryotic transcription and genes. In prokaryotes, when RNA polymerase bound with DNA, it will be lead to transcription, and transcription and translation can be take place simultaneously because absent of nucleus and absent of nuclear membrane and not necessary exit of messenger RNA to cytoplasm altogether, and all can be take place simultaneously. For eukaryotes, all transcription take place in nucleus, and in nucleus will be synthesized pro messenger RNA, which will be consist exons and introns, and necessary to provide splicing, excision of introns, and next modifications, for example, it capping, binding of cap on 5' end, and polyadenylation on 3' end. It's important for storage for stability of messenger RNA in cytoplasm. Later necessary exit, and after it will be synthesis of protein by translational machinery. And important differences between pro and prokaryotic and eukaryotic transcription, it mechanism of activation. For prokaryotic, RNA polymerase bound with promoter, and if place between promoter and coding sequence free, RNA polymerase will be go ahead. Not necessarily specific activities for its activation. Sometimes maybe activate the proteins, but it not influence dramatically. RNA polymerase bound and go ahead. For eukaryotic transcription, all of it much more complex. Binding of RNA polymerase with promoter activity, you can see TA box there, and not absolutely not necessary lead to transcription. For it necessary multiple activation factors, transcriptional factors which can be facilitate, activate, to provide enhancement of transcription of its gene. And if it transcriptional factors, activation factors, will be absent, transcription will be absent or will be very slow. It very important differences. Prokaryotic every time ground state on, switching, and only if necessary it will be preventing of it. For eukaryotic, ground state off, and necessary for activation, necessary a lot of factors. Next detail for prokaryotic gene organization. It can be organized in operon principle, operonal model. Is all genes organized? Not, but near half of all genes in prokaryotic genome organized in operonal type. Especially true for plasmid genes. It promoter, one regulative part operator, and a lot of structural genes. All transcription of all genes, structural genes, it called other name, it's cistrons, regulated by the same promoter, and all genes will be coded, transcribed by simultaneously. And later, messenger RNA will be template for multiple proteins. All or nothing. In eukaryotic genomes, one promoter will be regulate only one polypeptide, one messenger RNA, which will be coded one polypeptide chain. Excellent operonal type of regulation. Так, and movement of RNA polymerase go along DNA with melting place necessary to dissociate of DNA upstream of RNA polymerase, recognizing and binding of promoter, but in eukaryotic, not only eukaryotic, in DNA, double strand must be rewinded. And for it necessary, like in replication of DNA, it topoisomerases, enzymes, which will be unwinded, provide unwinding of DNA for avoiding of supercoils, which will be prevent of transcription. And again synthesis of DNA and RNA, now we tell about RNA, again possible only from 3' end, elongation on 3' end, and synthesis every time take place from 5' end to 3' end. Why? Because source of energy for its synthesis will be provided by nucleotide 3-phosphate, and for synthesis of RNAs necessary set of nucleotide 3-phosphate, it ATP, GTP, UTP, and CTP, like DNA synthesis. And now again topoisomerases, it enzymes which will be hydrolysate polynucleotide strand before RNA polymerase, unwinded of it. Again, it topoisomerase is possible divide on two types. It first and second. First, it will be digest double strand simultaneously. First type, it digest one strand and cleavage and re-synthesize. It nucleases really, and it lead to possibility of unwinding and prevent of supercoils. Well, and about RNA polymerase. RNA polymerase, now you can see it multi-subunit structure, and important structure, important part of it polymerase, it's so-called sigma subunit. Sigma subunit, it really promoter recognizing structure. It moved along DNA, and when we will be to meet of promoter site, it will be bound, and later it will be signal for association of all RNA polymerase, and later it will be lead to movement of RNA polymerase along template strand. Again, sequence of promoter -10 and -35 nucleotides upstream of coding path. Again, synthesis not go across immediately synthesis of messenger RNA. RNA polymerase gradually try to adaptate, and sometimes can to synthesize. Initiation can be beginning and sometimes stop. Again, beginning, stop, and will be abortive initiation, and later after association of all components of RNA polymerase, it will lead to at last will be beginning synthesis. It's you can to see for synthesis of proteins used a lot of energy. It's not necessary to keep energy storage it for any other. Protein synthesis used a lot of energy, and multiple factors and multiple variants of beginning and finishing of transcription. And now you can to see again template encoding strands, template strand, it main for synthesis, and it RNA polymerase of prokaryotes, which moved along template from 3 to 5' end. Synthesis of messenger RNA beginning from 5' end to 3' end. You see elongation of 3' end. For eukaryotic promoters, we talked about some more complex TATA box of promoter recognized by RNA polymerase, but regulative paths which localize upstream of coding path. Its regulative paths sequences can be localized sometimes upstream of some hundreds, some thousands, and 10,000 nucleotides of upstreams. Sometimes its regulative path can be localized on other genes, in introns of other genes. It's very complex and not every time good understandable variants of regulation. And we have three, in eukaryotic, in humans, 3 types of RNA polymerases. First type of RNA polymerase 1, it RNA polymerase which synthesize ribosomal RNAs. And not all ribosomal RNAs, but main it scaffold maybe to tell about basic structure of ribosomes. 27 says ribosomes, 18 says ribosomes, 5.8 ribosomes. Yes, ribosomes. RNA polymerase is main enzyme for synthesis of protein coding genes and small nuclear RNAs and microRNAs. Protein coding messenger RNAs and the regulative part of it for its genes. And RNA polymerase 3, it transfer RNAs and any components of ribosomal RNA. 5S RNA important for recognizing of start codons, by the way, important. And also any proteins which is important for function of small nuclear RNAs and nucleolar RNAs. You can to see, no, shortly, main protein synthesis messenger RNA is RNA polymerase 2, RNA polymerase 1, ribosomal RNA, RNA polymerase 3, transfer RNAs. And important for transcription, it for eukaryotic genes, it sequence must be free from nucleosomes, and it takes place competition between transcriptional factors and and histones. If histones bound with DNA, it will be lead to preventing of interaction of transcriptional factors, but competition depends on sequence and depend on situation in this region of genome. We talked about acetylation, for example, of histones lead to less intensive compactization and possibility to interaction of transcriptional factors. And demethylation of nucleotides in genome will be lead to possibility more or less intensive transcription. And again, you can to see RNA polymerases. RNA polymerase 1, it ribosomal RNA, 2, it messenger RNA, pre, of course, and 3, it transfer RNAs. And now situation in promoter region of eukaryotes, it must be so-called TA box of promoter, and which will be recognized by RNA polymerase, and a lot of different factors which can be activate of transcription. All name of activator, it enhancers. Enhancers can be any, sometimes maybe more than up to 10 or sometimes more enhancers, which will be recognized and bound with different activated proteins. Sometimes it may be proteins, may be receptors of hormones, for example, steroid hormones, which will be facilitate or not. Sometimes it may be recognized by separated by insulators, silencers, which can be influenced by inhibition of transcription, and a lot of other responsive elements, total name it, responsive element, and the inner space of which will be also state of its situation will be influenced. Sometimes you can to see enhancers can be localized downstream of promoter region. Relatively complex, and in genetic engineering it will be necessary to use for regulation of its activity. Now you can to see example how it's possible to possibility of regulate of transcription by sequence which localized far from promoter region. It result of folding of DNA with decreasing distance between enhancer regions and promoter regions. And now you can to see participants of regulation, polymerase 2 and activation factors, elongation factors, activation factors, TAT box associated factor, and other TATA box binding protein. Now you can to see it complex. Now it only for understanding that it's complex. And transcription, it's a dangerous situation for mutation, because when will be appeared one strand DNA, it leads to increasing possibility of hydrolysis and possible spontaneous hydrolysis by water phosphoester bonds. And in this case, it will be lead to stop of transcription block. In this case, it will be signal for stop of transcription, stop of activation. It will be lead to activation of reparation of synthesis, and but it reasons for appearance of double strand breaks, most dangerous, and reparation mechanism will be different. It may be or homologic recombination or non-homologic and joining. We talked about it on previous lecture. And it leads to activation of proteolytic digestion of proteins, which will be lead to stop of transcription. No, at first, totally, it dangerous state for a dangerous reason for mutation. Now you can to see elements which can regulate a real situation for different proteins. For example, gene which can regulate of immune answer in pregnancy. Uteroglobin in period of implantation of morula in first step of development of organism, embryonal development, necessary to prevent immune answer against new embryon. And for example, uteroglobin, it's important factor for it. And you can to see promoter region, and in promoter region consists a lot of factors place which will be regulated. And for example, one of enhancer localized on 1000 nucleotides upstream of coding path. And next, a very interesting variant of regulation of lactase, gene of lactase. Lactase in human population, mammals totally, for its synthesize up to 5 years old, 3-5 years old. Later, not necessary, because feeding by milk practically absent. But in any population, for example, in Europeans and any African population and Arab populations, which can drink by fresh milk, take place mutation in enhancer, and enhancer will be influenced on promoter region of lactase gene, and this persons in this population can to use fresh milk without any problems. But for other humans, using of fresh milk will lead to diarrhea and other problems, because lactase will be absent, lactose will be present in intestine, and it will lead to osmotic and any other non-pleasure consequences of using. But for example, any populations, in Kenya, Tanzania region, mutation take place in 14,000 upstream, and Arabian, it other place of mutation. You can to see changing guanine to cytosine, in this place in Arabian population. Thymine to guanine. In Europeans eat cytosine to thymine 13910, and in Ethiopia, Sudan region, it's also other. All of its mutational factors localize in 12 exon of other gene. You can to see MCM6, it mini chromosome maintenance gene, which will be activate of DNA replication. Why? No, you see evolution localize it regulation part on other gene. You can to see when very original place. Well, and now will be a short break, some 7 minutes, please. Well, dear students, will be continue. And next step of transcription, it excision of introns, splicing, total splicing. And you can to see after synthesis of messenger RNA, pro-messenger RNA, RNA will be consist introns and exons. We talked about average number of introns near 9, and it must be excited. And for it, excision provided by, now you can to see example in electron microphotograph, microshoot of microphoto. Yes, of it when it was first time evidence of it situation. Sequence of introns, in introns present specific sequence with GT in DNA, GU in RNA, and AG at the end of introns. And in the inner space must be specific sequence with essential adenine. And digestion, beginning from forming of, now you can to see, yes, GT in DNA, GU, AG, GU, AG in RNAs. It interspecies place, and will be beginning at first splicing of first splice site, digestion of hydrogen phosphorester bond, and forming of covalent bond with second carbon of ribose in adenine, this conservative sequence. Forming of lariat, and later digestion second phosphorester bond, and forming of two digested part, which will be spliced. For it, in this reaction participate small nuclear RNAs. Small nuclear RNAs in complex with proteins U1, U2, U3. It factors which will be bound and facilitate of its interactions. Sometimes small nuclear RNAs localize in inner space of introns. It introns by himself can facilitate of its excision. Now it some tens up to some hundreds components can participate in this forming of splice, so-called splicemosome, very big. Now you can choose the largest, but maybe not one of largest cellular machines. And splicing, it reasons of multiplicity of variants of proteins from one gene, because in different tissues, in different cells, can be realized different variants of splicing. For example, you can to see it tropomyosin gene will be in striated muscles, smooth muscles, in fibroblasts, and other in brain muscles. In brain, messenger RNA can be realized different variants of combination, different combination of exons, and it will lead to different properties of polypeptide chains, correspondent products. Sometimes maybe alternative variants of functions. For example, in cells synthesize one of components of regulation of apoptosis, so-called BCL gene, genomic locus, which will be a combination of different exons. But second exon can be can consist one additional part, and if it additional part will be absent, in this case will be realized apoptotic variant, pro-apoptotic variant of product. Activation of apoptosis, cell will be died. But if will be present additional factor which will be present in the second exon. In this case, activity will be anti-apoptotic. And its variance can lead to alternative activity. You can to see it, important for function. Next important and essential modification of genome, but it modification of DNA level. It forming of polypeptide chain in immunoglobulins. Immunoglobulins consist it's symmetric molecule which consists heavy chain for domains and light chains of 2 domains. And combination of heavy and light chains in antigen binding site consists individual separate sequence in combination of amino acid radicals which will be specific for recognizing of correspondent antigen. And combination of multiplicity of combination depend on multiplicity of reorganization, recombination of genetic material. Any minutes ago we talked about length of genes of heavy and light chains of immunoglobulins. It consists pieces, places, elements which can different number of repeating but different in a sequence of DNA path. It so-called V path which consists some hundreds variants of pieces. D path in heavy chain, some tens up to 15, and J path, some elements. And in period of maturation of genome in B lymphocytes. No, by the way, immunoglobulins it soluble receptors of B lymphocytes. And different combination when, for example, first from some 100 variants, first V, first D, first J will be lead to variant of 1. The same for a light chain. It can be different, some hundreds of V variants, D variants absent, but J also some units of paths, and different variants of recombination will be lead to multiplicity of variants of variable region of immunoglobulins. And additionally, a constant region also variant of mu, delta, gamma, different variants of gamma, epsilon and alpha paths, and association of variable region of immunoglobulin gene with constant region lead to forming of whole molecule of gene of whole molecule of immunoglobulins. Now the same mechanism for T cell receptors, T lymphocyte receptors. Also multiple variants with but T lymphocyte receptors, receptors constantly bound with membrane. B lymphocytes consist soluble receptors or immunoglobulins. T lymphocytes consist constantly associated receptors in membranes. Next variant of maturation of messenger RNA, we told it capping. Capping it association of methylguanidine by associated non-usual, not phosphoester bond between ribose and phosphate, but any 3 residues of phosphate associated one to other. non-usual, and it will be stable for nuclease activity in 5' end. It modification of 5' end. And it methylguanidine, additional methyl group will be again. And defense in 3' end by polyadenylated tail. Also non-coding part upstream and non-coding part downstream of messenger RNA. Totally, you can to see not only it, but also consists additional signals for binding with ribosome. CAP, methylguanidine on 5' end, later specific hairpin loop, stem loop hairpin structure, which recognized by ribosome and facilitate of interactions. So-called IRIS site, it internal ribosome entry site, which also additional factor for binding with ribosome. It all of it 5' uncoding sequence, uncoding non-translated region. Sometimes it can some hundreds nucleotides, up to some thousands. Coding part. And later again 3' untranslated region or downstream of coding part, which can be also consist specific hairpin structure, loop, terminal loop, which will be indicate about finishing of gene and its signal for dissociation of ribosome. And sequence which will be regulate subcellular localization and provide stability. It usually polyadenylate tail and some specific sequence which will be also recognized and prevent of 3' nuclease activity. And next step after preparing of messenger RNA, beginning next translation. Mechanisms similar, but initiation can be different. But all of it consists of the next step. It pre-initiation or so-called activation of amino acids, namely real translation. It consists initiation, elongation, and termination steps, and post-translational modification. And some hundreds peptides and proteins participate in this. All of it so-called housekeeping gene derivates. Housekeeping gene which constantly synthesize. And first step is activation of amino acid. Every time any polymerization in living organism take place, go across intermediate substance, high energy substance must be activated, monomer must be activated. No, for example, synthesis of glycogen, it's not free glucose but UDP glucose. Also the synthesis of amino acid, polypeptide chain, amino acid must be activated. Macroreactive substance which called aminoacyl adenylate. For it, ATP will be interact with amino acid and will be interaction of phosphate group of AMP, pyrophosphate will be digested, and by phosphate AMP will be interact with carbon and carboxyl group of amino acid. And later it will be lead to interaction with transfer RNAs. About it, amino acid, you can to see carboxy group and ATP minus pyrophosphate and binding of phosphate with carboxy end. And later interaction with transfer RNA. On transfer RNA, transfer RNA consists two ends, 5' end like any RNAs, 5' end and 3' end. and interaction of aminoacyl adenylate, macroalgic aminoacyl adenylate, will be bound with 3' end ribose or second or third carbons. We have two types of enzymes which will be provide association of aminoacyl adenylate with 3' end of tRNA. First type, which will be interact with second carbon OH group, and second type will be interact with third carbon. It very conservative ancient enzymes with long time evolution, and storage of primary structure of it very important. Now about structure of tRNA. tRNA is relatively short, up to 85 nucleotides, with a very specific sequence with inner palindrome sequence which lead to forming of stems and loops. And the three loops consist. One loop consists anticodon, which will be specific for each tRNA, for each amino acid. All tRNAs consist on 3' end, the same sequence C, cytosine, cytosine, adenine, and to ribose of last ribose of 3' end will be associated active aminoacyl adenylate. And two loops, one of it will be dehydrouracil loop, D-arm, and pseudouridine loop with so-called pseudouridine loop. And sometimes may be present more or less long variable arm. And in three-dimensional space, tRNA folded in L-like in Latinus or G-like in Kirillitsa letter, with 3' and ready to bound with aminoacyl adenylate, and anticodon with the possibility of binding with messenger RNA in translational pathway. The sequence of other nucleotides also can be very important. For example, now example of transfer RNA of mitochondrial RNAs, and must be modification of nucleotide radicals usually by methylation. If methylation or its modifications will be imperfect by defects of corresponding enzymes, it will lead to serious dysfunction of cells. It will lead to serious encephalopathies. You can see defects mitochondrial tRNAs will lead to serious diseases. All of it dissociated with dysfunction of central nervous system. Deafness, cardiomyopathy, lactoacidosis, myoclonic epilepsy, mitochondrial encephalopathies. Mainly every time central nervous system will be participating in this. You're going to see molecular reasons of disease. Next, important for correct binding of amino acid with correct tRNA. For it necessary to know so-called genetic code. What structures know genetic code, what structures provide correct interaction of correct amino acid with correct tRNA. It provided by tRNA synthesis. tRNA synthesis, we told about it, two types. One of it interact with third carbon of ribose, and second type second carbon it first type, to third carbon it second type. And it you can see enzymes with complex with tRNA, which recognize and bound of amino acid. This is one. with 3' end and specifically recognizing part it anticodon loop. It two types of synthetase. Class first interact amino acyl group to second carbon group, second class to third carbon group. And about genetic code. For it correspondence between sequence of nucleotides and sequence of amino acids. And now you can to see it cytoplasmic genetic code and some details. Oh, by the way, it was determined relatively quickly. First idea that was when Crick proposed about presence of it code. It was idea very long, but it was determined quickly, relatively quickly. And about details of it and properties of its genetic code. At first, it triplet, one amino acid coded by 3 nucleotides. And variants of triplets, combination of 4 nucleotides by 3 nucleotides triplets, possible 64. But not all variants important for coding of amino acids. 3 triplets senseless. It's called stop codons, UAA, UAG, and UGA. And other variant, it comma-less, unambiguous, and degenerate. Degenerate code, it tell about more than one triplet coded one amino acid. For example, you can to see phenylalanine coded by 2 triplets, leucine coded by 6 triplets, and isoleucine 3 triplets, valine 4, serine 4, proline 4, threonine, alanine 4. Sometimes you can to see more than one triplet, usually coded one amino acid. Only 2 amino acids coded by single triplet. It AUG by methionine and UGG tryptophan. Other usually 2 or 4 and sometimes 6 nucleotides by leucine for leucine. It's called degenerative code. And sometimes you can listen universal. But now it's not universal. It present some variants of genetic codes. For example, our mitochondrial genetic code, it have differences from cytoplasmic, but its difference is not too high. Talk about degeneration of codes. For example, serine important, first 2 nucleotides important, third not important. How it possible to describe? It described by Wobble hypothesis, because in translation synthesis take place adaptation of anticodon sequence of transfer RNA with more effective interaction for first 2 nucleotides. Third nucleotides is not such important. And now it variants of genetic code with mitochondrial variants in human mitochondrial DNA. Only 2 stop codons. For example, stop codon for cytoplasmic code UGA, for mitochondrial code is tryptophan. Tryptophan consists, you can to see, in mitochondrial code consists 2 triplets. Also, any other variants can be sometimes methionine, also maybe other variants. It tells about possible variants of modifications of code and evolution of it. And cytoplasmic code more convenient for constant working. But mitochondrial genome more conservative, and it reflects maybe more ancient variants of genetic code. Next important variant, AUG, or every time translation beginning from codon AUG. It methionine codon, and it's so-called start signal for synthesis. But for example, if you have AUG, but in different place, in different positions, what AUG will be correct? How it possible to determine? If first, in this case, second AUG will be part of other codons. If second, how it possible to exclude it of first? It depends on sequence upstream of start codon. It must be so-called in prokaryotes Shine-Dalgarno sequence, specific sequence which will be specifically indicate where start codon. For example, now it start codon there, and upstream sequence indicate about there. It result of physical association of upstream part of messenger RNA with small subunit of ribosome. Yes, position of ribosome at correct site will be true. Next, necessary to have ribosomes. What is a ribosome? It organelle will consist two parts, big and small subunits. But really it consists ribosomal RNAs and some dense proteins. And in free state, ribosome will be dissociated. And ribosomes can be free, which can synthesize soluble proteins, and fixed ribosome, which will be associated with membranes of endoplasmatic reticulum. Now with bacterial ribosome, size of it so-called 70S, and eukaryotic ribosomes 80S. Big subunit 50S in prokaryotic ribosomes, and 30S for small subunit. Totally 30 and 50 will be lead to forming of 70S ribosome. And for eukaryotic, 40S small subunit, 60S big subunit together will be lead to 80S. Why it not arithmetic sum? Because S it Svedberg units, it named by author of creation of first ultracentrifuge, Svedberg, and it hydrodynamic characteristic of substance. Because if will be compared sedimentation of spherical particle and ellipsoid, ellipsoid will be with the same molecular weight. Ellipsoid will be moved more quickly because hydrodynamic resistance for it from environment, water environment, will be less than for spherical particle. Shape and size will be important for sedimentation. And when 70S and 50S of prokaryotic ribosome will be associated together, it will be lead to more compact structure than sum of it. You can to see. And it consists, big subunit consists 23S ribosomal RNAs, small subunit 60S ribosomal RNAs, plus 5S ribosomal RNA in big subunits, and more than some 36 constantly associated proteins in big subunit, and 21 constantly associated proteins with small subunit. In eukaryotic ribosomes, number of proteins more and number of ribosomal RNAs also. But in eukaryotes also present 70S ribosome because mitochondrial ribosomes consist 70S. It derivates of ancient intracellular parasites. And a big subunit of eukaryotic ribosomes consists 70S RNA, 5.8 Svedberg RNAs and 5S RNAs. And a small subunit, 80S ribosome RNAs, ribosomal RNA, and up to 30 proteins. Ribosome consists, pro and eukaryotic, consists for binding sites. One site is binding with messenger RNA, and 3 sites for transfer of tRNAs. It intermediate P site or peptidyl site, A site amino acyl site, and E site exit site. And the ribosomes can bound with endoplasmatic reticulum with the possibility of entrance of newborn polypeptide chain to cavity of endoplasmatic reticulum in systems of endoplasmatic reticulum. Synthesis of ribosomes take place in nucleolus, and synthesis take place by single very long, no, not very long, but long pre-ribosomal RNAs. It's 45 as ribosomal RNAs, which later will be digested on components on 28th and 80th ribosomes and reconstructed of subunits by association with ribosomal proteins, which will be synthesized in cytoplasm and later necessary to come to nucleus and nucleolus. Nucleolus you can be described and observed in electron microscope, you can see there. And the separation and subunits after collection combination with proteins, and for example 5S ribosomal RNA important for function for initiation. It must be synthesized separatively and later will be associated with maturated ribosomes. Details of protein synthesis. Messenger RNAs moved and translated from 5 to 3' end, and synthesis of amino acid sequence will be beginning from amino end and finishing by carboxy end. And in ribosomal cycle present 3 phases. Initiation, beginning from first tRNA with codon AUG, elongation, binding of next tRNAs with next triplets, and termination, when stop codon will be achieved, maybe messenger RNA in ribosome will be achieved stop codon in messenger RNA. And again, I'll repeat, one promoter necessary for synthesis of operon in prokaryotic organisms, but AUG and will be synthesized all messenger RNA total for all peptides. But synthesis and beginning of binding with ribosome beginning in individual place of each gene. For it present special AUG, and in spacer will be a place for activation of translation, activation of binding of ribosome and activation of translation. Next, we talked about 3 sites in ribosomes, and beginning of transcription of translation beginning from association with initiation factors. Small subunit will be bound with the initiation factors. In prokaryotes, initiation factors 1, 2, and 3. But initiation factors with formylmethionine tRNA, initiational transfer RNA, bound after interaction of messenger RNA with ribosomes and appearance of initial codon in P site. It provided by interaction at first initial factors bound with 3 with small subunit, and second factor of initial bound with the formylmethionine transfer RNA and GTP. It energy-dependent process. Later, messenger RNA with the Shine-Adler-Harner sequence, which will indicate where is correct AUG, start codon, bound with the small subunit, and in this place with the Shine-Adler-Harner sequence will be recognized bound second initiation factors, which will be transport RNA to P site of ribosome. And later, after reconstruction of its initiational site in P site, big subunit will be associated with the small subunit, and the initial factors will be dissociated. For it, used first GTP. And in P site will be initiational tRNA, A site will be empty, and E site will be empty. And next will be beginning elongation. For elongation, necessary 2 factors with GTPase activity. In contrast with synthesis of in metabolic pathways, in protein synthesis participate mainly guanosine 3-phosphate, GTP. Only for activation of amino acids used ATP, but everywhere to GTP dependent. It lead to impossibility. It lead to absence of competition for macroergic substance between protein synthesis and other anabolic reactions in cells. And for it necessary elongation factors for prokaryotes, elongation factors. For eukaryotes, it will be E elongation factors, eukaryotic. And factors for modifications and GTP metabolism, it elongation factors T temperature unsensitive and elongation factors temperature sensitive. All of it with GTPase activity. And the peptide bond will be forming when P site will be bound with the next amino acid. And for translocation, necessary GTP, next one. It provided by next. In peptidyl site bound initial codon, A site will be free. And selection of correct tRNA will be bound with next A site. For it necessary factor elongation with GTP. Binding of elongation factor and next amino acid tRNA lead to hydrolysis of GTP, and it lead to forming of ester bond between previous amino acid and next amino acid associated with elongation factor. It provided by RNA, ribosomal RNA, immediately after it. Later, elongation factor G in prokaryote will be displaced of previous tRNA associated with P site, with A site, and displaced to P site. Ribosome will be moved on one triplet, and for its activity necessary elongation factor G associated with GTP. Or in eukaryotes, it elongation factor 2, very important. Without it, movement ahead will be absent. And after it will be elongation factor will be dissociated. In P site will be amino acid which will be previously associated with A site. Next, tRNA without amino acid will be dissociated from E site. And next, elongation factor associated with next GDP and next amino acid will be associated with A site, empty A site in next step. And it will be elongate. Now you can to see elongation factor G or elongation factor 2 on eukaryotes have similarity in structure like amino acid and elongation factor. Second, similarity with possibility of binding with A site. Altogether lead to, you can to see movement of messenger RNA, now with P site, A site and E site, but it's not simultaneously associated with tRNAs. Direction of movement of messenger RNA and place of synthesis of polypeptide chain from amino end. And later, when will be appeared empty place in A site, stop codon, one of 3 stop codons, will be binding of releasing factors, TF terminal factors or RF releasing factors, again GTP necessary for each association, and digestion last amino acid with last tRNA associated with P site. And later it will be lead to dissociation of ribosome. Now you can to see releasing factor 1 bound, releasing factor 3 will be lead to stop and dissociation of ribosome. One messenger RNA can bound with multiple ribosomes with forming of polysomes. And bioinformatic with investigations of evolution of sequences can lead to some interesting fact, can lead to reconstruction of ancient sequence of elongation factor 2, and it will be lead to possibility of structure which was a long time, 3.5 billion years ago, for first living organisms which will be appeared on our planet, and synthesis of polypeptide chain which correspond of its elongation factor, and the determination of most active in what temperature it's most active, led to 3 independent pathways led to the same result. It most optimal temperature of its activity near up to 70 degrees of Celsius. It tell about ancient temperature which was 3.5 billion years ago, when life was present, but temperature will be temperature was high. It's all about gradual decreasing temperature of our planet. Now optimal temperature for this now present living organism, for example, E. coli, near 37-40 degrees, and 3.5 billion years ago, most optimal temperature was near 60, 65, up to 70 degrees. And for synthesis of all polypeptide chain, for each one amino acid in polypeptide chain, necessary minimum 4 equivalents of ATP for GTP, plus 1 ATP for activation of amino acid. Now regulation of translation possible by activity of enzymes which work for synthesis, possible by frequency of initiation, number of messenger RNAs necessary for synthesis of eight polypeptide chains, and the abundance of ribosomes. And the frequency of association with ribosomes depend on upstream, 5 upstream path. And any viruses used internal ribosomal entry sites without capping it enough for competition between viral RNAs and ribosomes. For example, iris of any viruses, hepatitis C virus, it effectively lead to effective competition between cytoplasmic oven messenger RNA and viruses. Next, phosphorylation of elongation factor 2 and other factors. Very often it can be antibiotics, but about it we will tell on next lecture. Thank you very much. Goodbye.